Welcome to the fourth part of the PamGene data analysis training. In the previous videos, I talked about experiment design, image analysis, basic processing, and phosphocyte analysis. In this video, I will talk about kinase analysis. Here is an overview of the downstream parts of the two types of workflows. In the C1T1C2T2 design, we are interested in comparing test versus control in each supergroup, and also sometimes we compare the same conditions between the two supergroups. In the C1T2T3 workflow, we are interested in comparing multiple conditions versus one control condition. For example, everything versus control, T2 and T3 versus T1, and T3 versus T2. You can see the Upstream Kinase Analysis apps in green rectangles and that they are after combat correction. Let me start by explaining the background of the Upstream Kinase Analysis. The Upstream Kinase Analysis is a functional class scoring method, meaning that to predict a kinase, it analyzes the changes in its set of phosphocytes. A phosphocyte set of a kinase comes from public databases of kinase protein phosphocyte relationships. In functional class scoring, scores are assigned to functional classes, indicating how strongly the kinase is associated with the observed changes in peptide signal. We run UKA on all QC-passed peptides, not only on significant peptides, thus it is a parallel to phosphocyte analysis. The output of UKA is similar to the output of phosphocyte analysis. It is the fold change and significance of kinases changed between a test and a control condition. There are different types of plots available, like volcano plots, bar plots of the top kinases, and coral tree, which also shows the family relationships between kinases. Here is how the Upstream Kinase Analysis algorithm works. On the left side, in blue, we can see the known phosphocytes of a kinase, kinase 1. This information comes from public databases. On the right side, in orange, we can see the phosphorylation data resulting from the PamGene experiment. We have information of what substrates have changed in test versus control and how they changed. Red arrows pointing up indicate a positive fold change. Blue arrows pointing down indicate a negative fold change. The thickness of the arrows indicate the size of the change. Next, for each kinase, we overlap its known phosphocytes and the phosphocytes in our data. Here, we had 3 overlapping phosphocytes for kinase 1. Based on the fold change of the phosphocytes, the algorithm calculates the fold change of the kinase. Next, we have to understand how likely it is that these kinase fold changes are real. Which kinases have really changed in the data? To assess this, we ask the question, could our data produce the observed kinase fold changes just by accident? For this, we make use of random permutations of the data, and we assess whether the observed fold changes are different than random. This likelihood of whether the kinases have really changed is measured by 2 scores, the significance score and the specificity score. To calculate the significance score, the samples are permuted 500 times, and the kinase fold change is recalculated in each permutation. If samples are permuted, we calculate phosphocyte fold change between samples which are randomly assigned to test and control. Therefore, if the kinase is truly changed, its absolute fold change will be much higher than the fold change coming from the random permutations. Here you can see that for one of the phosphocytes, the fold change even changed the direction. The significance score is calculated based on the difference between the actual kinase fold change and the kinase fold changes coming from the permutations. A high significance score means a high probability that the kinase is differentially active between test and control. The second score for assessing whether the kinase is significantly changed is the specificity score. For significance score, the samples were permuted, and for specificity score, the phosphocytes are permuted between phosphocyte sets. Thus, it is not our actual observations that we permute, but the database. If phosphocytes are permuted, random peptides will be assigned to kinases. If a kinase is really changed between two conditions, it is unlikely that random phosphocytes will give similar fold change. The specificity score is calculated based on the difference between the actual kinase fold change and the kinase fold changes coming from the permutations. So, we have two scores, the significance score and the specificity score. As p-value, we use the sum of these, which we call the final score. Let's take a look at the output file that we get from the Upstream Kinase Analysis. The first column is the test condition, which is again a comparison column. Here it would be probably T1 versus control. Then we have the kinase name, and there are some other annotation columns that I didn't show here, for example, the uniprot. The median kinase statistic is what we use as fold change. It is actually the fold change scaled by the noise, and the actual fold change is in the kinase change column. Next, there is the significance and specificity score columns, and the final score, which we use as p-value. Our usual threshold for final score is 1.3, which is equivalent to a p-value of 0.05 on minus log 10 scale. You can also observe that all these scores are actually mean or median scores. It is not a must to understand this, but if you are interested, please read about it in the PDF called UCA, Details of Algorithm. Next, let's look at the workflow overviews again and talk about the placement of the UKA apps. They are after combat correction. However, for combat, we have to keep in mind that it should only be done if the conditions are balanced over the technical batches and if there are batch effects and combat can correct them. Otherwise, this step can be just deleted, and the UK steps can be linked to the log or VSN steps. Finally, before we go to Tercen and I show how to run the Upstream Kinase app, here is an overview of the different UKA apps. Based on the experiment design, we run different UKA apps, but the core algorithm is the same. In the CT1, T2, T3 templates, we test multiple test conditions against a single control condition, so the app is named as UKA_NTVC. However, there is no similarity with the algorithm in the NTVC app for phosphocyte analysis. In the C1T1, C2, T2 templates, the UK app is called the UK2 Group Comparison, or TGC app. It compares one test condition to one control condition, but it can do it in multiple supergroups at once. Now, let's go to Tercen. In the CT1, T2, T3 workflows, we have both the UKA and TVC and UK TGC apps. Only the T3 versus T2 comparison has to be run with the UK_TGC app, since the UK_MTVC app always expects multiple test conditions. You can't really see which app it is, but they have different version numbers. For these comparisons, we pre-filter the test conditions. This is not like how we do it in phosphocyte analysis, MTVC, because there the ANOVA takes all comparisons into consideration. Therefore, we do not pre-select the conditions, but later filter for the interesting ones. However, the UK works per comparison, thus pre-selection of interesting conditions is needed. For example, here, we want to do a UKA with the control condition being T1. So in the step before, we filter out control and put the identity operator. Next, let's see how the wizard of this UKA app looks. Almost all questions are pre-filled, only the quantitation type is needed. There are two filters for this app, the selection of the control and the test conditions. The easiest way to select test conditions is to filter for test condition not equals control. However, notice that the selection is needed to be changed to T1. Regarding the quantitation type, can you guess what it would be? It is called identity from the previous step with the identity operator. Let's check the results of this UKA app that I have run before. Double-click, and we can see that there is one visualization. the volcano plot, which is very similar to the ones we have from phosphosite analysis. Instead of the p-value, we have the median final score on the y-axis, and instead of the fold change, we have the kinase statistic on the x-axis. The median final score is colored in a way that above 1.3, the usual threshold, it is red. There is also a fancier volcano plot available, but since this is a shiny app, it doesn't show up in the visualizations. So we have to go inside the app. The crosstab view of this shiny volcano is almost the same as the simple volcano. There is one important difference. The kinase name is in the labels. The labels identify each observation. Let's run it. Here we can select for the condition we want to see. Next, we have to set the thresholds to be suitable for the UKA. The default for the fold change threshold is quite high. It is very common that even kinases with very high scores have low fold change. So I set it to, let's say, 0.3. Next, let's set the significance threshold for 1.3. Here, we can also set how many kinases we want to see by name. Or, if we want to see where specific kinases are, we can select those from the dropdown. The Shiny Volcano app knows these names because we have put the kinase name in the labels. Next, I will show how to make a UKA bar plot. This you can even do in Excel easily, from the UK output file. The UKA bar plot works for a single condition. So before the bar plot, I selected the T1 condition. It has to be selected in this way. All the factors we want to see in the bar plot have to be in the rows. Let's run the app. In the wizard, everything is pre-filled correctly. The same app has been run here, let's check it. This figure is made by the plot operator. You can see that the bars show the fold change of 10 kinases. Let's reset to see how it works. These 10 kinases are the top kinases according to the median final score. This ordering is done in the previous step by the order by operator, which we have seen before in the simple heat map app. In the plot step, there is a filter for row order, which you can customize. The bars are colored by the specificity score, and you can customize this as well. This step has the plot operator, which makes fancy plots. You can play with the settings. Here I set the names of the axes, the output file extension, the width and height in pixels. The flip parameter switches the x and y axis. Let's run it. Below the plot, you can download the figure. You can also make bar plots colored by kinase family. You can just exchange the family for the specificity score in the wizard. There are a lot of kinase families, but only 7 main kinase groups. There is a type of figure that shows the family relationships of kinases within these main groups. It is called Coral Tree, and it is a public website. You can find the link in the PDF or just search for Coral Tree Kinome. The info section walks you through the settings. Let's go to the plot section. This is the basic coral tree showing the 7 families. TK is tyrosine kinase, TKL is tyrosine kinase-like, and the rest are SDK families. This tree can be updated by our data. We use the node color for median final score and node size for kinase statistic or fold change. We have to get the data from the output file, so let's go back to our project. The UK output file is in the exported data folder. Let's download it and open it in Excel. Let's filter for one condition, T1. Then, order the rows by median final score. I will make a new sheet to copy the first 20 lines, but I suggest that when you do this, you copy the data from both the PTK and SDK output for the kinases with median final score above the 1.3 threshold. I copy the uniprot identifier, median final score, and kinase statistic columns. Then I'll copy first the uniprot and the kinase statistic, open the Coral Tree app, and paste it to the node color. We have to set the scheme to quantitative, and we also have to set the identifier to UniProt so that it is recognized. You can see that the nodes appeared. Then, let's copy the UniProt and the final score to the node size. Importantly, I recommend that in the advanced settings to set the label identifier to HGNC. This is the same as the UniProt primary gene name. This is what we use as well for kinase name. This concludes the Upstream Kinase Analysis. You have learned about how the UKA predicts kinases and what the different scores tell about the significance of the predictions. Insight into signaling pathways and networks can be interesting. The next training part will provide guidelines on this topic. If you would like to validate your results, here is a short guide about what to and how to validate. You can stop the video here to read, or you can find this slide in the PamGene Data Analysis Guide PDF. Do you have any questions? Please contact us.